This session is related to the paper titled Optical Instrumentation and Optometry. Myself, Prakash, working as an assistant professor in the Department of Physics and Electronics, K.B.N College. So, in this video, we are going to see about the first unit, which was titled as Optical Microscopes. Actually, this paper titled Optical Instrumentation and Optometry consists of different units like basic optical microscopes, what are the optical instrumentations, and the third unit related to the applications. Like this, all these things are related to the optical instrumentations and the optometry, which we are using in the daily life. So, in this regard, the first unit is titled as Optical Microscopes. So basically, we have different types of optical microscopes like simple microscope, compound microscope, traveling microscope, phase contrast microscope. We have different types of microscopes. Depending upon the specimen, we'll use the different types of microscope. So in this session, I'm gonna have to cover, like, first we'll see what is simple microscope. What is the construction, how it works, and what are the applications that a simple microscope will have. And then we have about the compound microscope, working principle, how the construction is made, and applications of compound microscope. Then we'll see what is how a simple microscope will differ from a compound microscope, and then about the traveling microscope. So first of all, a simple microscope. A simple microscope is a basic optical instrument that will magnify the small objects. Actually, the term microscope will itself says that it magnifies the object what we are seeing. But the simple microscope means it uses a single convex lens or a set of convex lenses in order to magnify the image. But apart from the compound microscope and traveling microscope we have, the simple microscope will works like a magnifying glass. It just works like a magnifying glass. Okay. Coming to the construction of a simple microscope will majorly have 8 parts. Like the first part is the eyepiece. The eyepiece is the lens that is closer to the eye, which we could able to see the specimen. Okay. And it helps us to view the magnified image of the specimen, the eyepiece. The second one is the nose piece. It is just like a rotating holder which will have the objective lenses. The nose piece will have the objective lenses. Then the objective lenses, the third part, the objective lenses, it will have different types of magnifying lenses, like 10 times the magnification, 40 times, 30 times. Depending upon the magnifying power we need, we'll use the different objective lenses. The fourth one is regarding the specimen stage, the stage where we will place the specimen and it will be seen from an eyepiece. So coming to the fifth part, we'll have like a light source. In order to see the specimen, we'll be illuminating the light from the bottom of the specimen. Then the light will fall on the condenser lens and it will fall onto the specimen, and the image that is formed from the objective lens is viewed through the eyepiece. Then the next one is the condenser lenses. The condenser lenses will focus as the light onto the specimen. Here we can see that when the light is switched on, the light will fall on this specimen stage. Then it will fall onto the objective lenses. From here we could able to see from the eyepiece. Okay, and the two mechanical parts that we have are the body or the arm. It will connect the base and the eyepiece part. So the connection between the base part and the eyepiece part is called as the body, or also called as arm. Then the last part is the base. So the base part is the base part will give the stability to the entire instrument. It will not make the simple microscope to tilt. It will give the stability and support for the microscope to withstand. Okay. These are all about the construction, what will have the different parts in a simple microscope. Then the working principle. The working principle behind the simple microscope is simple, like we can see here. Initially, that light will fall onto the condenser lens. Okay, when the light is illuminated, it will fall on the condenser lens, and the condenser lens will make the light to fall onto the specimen stage. Here where we'll keep the specimen, what type of sample we want, we'll keep on the specimen stage, and the light which is falls on this specimen or the sample is viewed by the objective lens. So then it is viewed by the eyepiece. Okay, this how it will works. So actually the simple microscope, light is refracted through a convex lens. Here we can see that the condenser lens is a convex lens. The light will fall on it will be converged onto the specimen stage. Okay, here the object is placed within the focal length of the specimen. The object should be placed within the range of, so actually these lenses will have some focal length, right? This specimen should have to be placed within the focal length of this convex lens. So after this process we could able to see a virtual, erect and an enlarged image of the specimen. Okay, from the eyepiece we'll see like the rays will be bent. Here you can see that the rays will be bent and they will be viewed like they are coming from a large image. Okay, the large image, they are coming from it will, as if it is coming from a large image, it will be seen from the eyepiece. So the next one is like magnifying power. What is the magnifying power of the simple microscope? So magnifying power is defined by the formula m is equal to 1 plus d by f. So where m is the magnifying power of the lens or the magnifying power of the simple microscope, and d is the least distance of distinct vision. So it is actually 25 centimeter for a normal human eye. Actually the least distance of distinct vision is the distance in which a person could able to see an object without feeling any difficulty. Without any difficulty, if you could able to watch that image, that distance is called as the least distance of distinct vision. Then f is the focal length of the lens that we are using in the simple microscope, nothing but the condenser lens. Okay. Next one, what are the applications we have? Coming to the applications we have, the simple microscope are mainly used in different fields, like in botany and biology laboratories, in order to view the insects, in order to view the wings of the insects, what they are studying. It is used there. And second one is the precision crafts. Like watchmakers will usually use a simple microscope for their working purpose, in order to see the internal working of the simple microscope. Internal working of the watches, they'll use the simple microscope. Then they are used in jewelry inspection. In jewelry inspection, they will see the fine details in gemstones and precious metals. They use the simple microscope in order to magnify the image that they are seeing. And the last one is a medical purpose, like in dermatology, in clinical work, they use it to examine the tissues in the skin cells. Like skin samples, they use it to see. They are used in medical applications. Next, about the compound microscope. So this itself is a compound microscope. This is how it will look like. So coming to the compound microscope, the difference between the simple and the compound microscope is that the simple microscope will majorly have a single convex lens, but the compound microscope will have different types of convex lenses, and it will have the different magnifying powers as well. So compound microscope is a powerful optical instrument which was used to observe the cells, microorganisms, transparent specimens, different types of specimens. Like the works with the two sets, it works like, as I said, it works with the two sets of lenses for the higher magnification. Compared to that of the simple microscope, in compound microscope will have the higher magnifications. It is used for more advanced applications than simple microscope. Coming to the construction, basically the construction of compound microscope is classified into two types: optical parts and the mechanical parts. Like which are related to the light and everything will be termed as an optical parts, and the which are regarding to the mechanical, like screws and everything, will come under the mechanical parts. The first part is called the eyepiece, as we have seen. So it is the part which is near to our eye, and it will magnifies the image from the objective lenses. Okay, it is there in the simple microscope as well. Then the next one is the objective lens. The objective lens is near to the specimen, and it forms a real inverted and magnified image of the specimen. Then the condenser lens. The condenser lens will also work as well as similar to that of in the simple microscope. It will make the light to focus clearly and contrast onto the specimen. And there are mirrors and light sources which will provide the illumination from the below part of the from the bottom part of the microscope. They will provide the light that is sufficient, which was required to see the sample. The next one, regarding the construction, these are all the mechanical parts we have, like body tube, revolving nose piece, coarse adjustment knob, fine adjustment knob, inclination joint, and the base part. So coming to the body tube, it maintains the proper distance between the lenses. Because the body tube will maintain some distance between the specimen and the lenses. The next part is the revolving nose piece. It will have different types of magnifying powers, like 10 times the magnifying power, 40 times the magnifying power. It will have different types, and it carries the multiple objective lenses and which are rotatable. We can able to rotate them. Okay. The next one is a coarse adjustment knob for quick and rough focusing. In order to have a rough focus, we'll use this knob called as coarse adjustment knob. And the next one is fine adjustment knob. Fine adjust knob is for the precise focusing. So coarse adjustment knob and fine adjustment knob both are different. So for the rough focusing, we'll use the coarse adjustment knob. For the precise focusing and slow focusing, we'll use the fine adjustment knob. Then the inclination joint. It allows the tilting for convenient viewing. For some samples, in order to view them, we need to tilt them in certain directions. Then we'll use this inclination joint. Then the base part. It will provide the support and stabilizes the microscope without falling or anything. It will provide the stability and support for the compound microscope. Then coming to the compound microscope, it involves, as I said, two convex lenses. In simple microscope, we'll have only one convex lens, but whereas in the compound microscope, we'll have two convex lenses. So the two convex lenses are objective lenses and eyepiece lens. So the objective lens will form a real inverted and enlarged image. First of all, the light will fall on the objective lens, and the objective lens will fall the light onto the specimen. Then what we could able to see from the specimen is the real inverted and enlarged image. This image, the output of the objective lens will become the object for the eyepiece lens. Okay, the output of the objective lens will become the object for the eyepiece lens. So from the eyepiece, we could able to see a real and enlarged image. Okay, just it like a simple microscope. So the final image will be real, inverted and highly magnified image. We could able to see an highly magnified image. So from this we could able to say that the simple microscope will have a single convex lens, but whereas the compound microscope will have a two convex lenses called as objective lens and eyepiece lenses. So similar to that of the simple microscope, compound microscope will also have the magnifying power formula determined by the m is equal to m naught times the 1 plus d by f. Here this part m plus d by f, if you could able to remember, this is from the formula of simple microscope, and this one m naught is the magnifying power of objective lens. MO or m naught will say. This is the magnifying power of objective lens, and this is the magnifying power of eyepiece lens. So in simple microscope we'll have only this part, but whereas in the compound microscope we'll have two parts like objective lenses and eyepiece lenses. So here we know that D is the least distance of distinct vision, and here FV is only the focal length of the eyepiece lens, not the objective lens. FV is the focal length of the eyepiece lens. So coming to the applications of compound microscope, similar to that of simple microscope, compound microscopes are widely used in biology labs, like for plant and animal cells microorganism examinations. They'll use this compound microscope, and in medical laboratories like disease diagnosis, for blood tissues like skin tissues, they will use this thing. And for the forensic science like crime investigation, trace analysis, in forensics also they will use this compound microscope. And for the material science and microelectronics study, they'll use these structures. And for the education and research purpose, a detailed microscope studies are used. So coming now we have seen about the simple and compound microscope as well. So simple and compound microscope will have these type of differences. Like coming to the lens system, simple microscope will have a single convex lens, but whereas in the compound microscope we'll have two sets of components like objective lens and eyepiece lens. That's why in compound microscope we'll have the formula like m is equal to m naught times 1 plus d by f. Coming to the image type, in simple microscope we'll have a virtual, erect and magnified image, but whereas the compound lens will produce as virtual image and an inverted image and highly magnified than the simple microscope. Next, the simple microscope is used for larger objects like tissues, insect parts generally, but whereas the compound microscopes are used for like cells, bacteria, microorganisms. In order to view them, we'll use compound microscope. The construction of simple microscope is somewhat simple, but whereas the compound microscope will be complex and it will have more mechanical parts compared to that of simple microscope. Coming to the applications, simple microscope is only used in watchmaking, jewelry, basic biology parts. But compound microscopes are used in forensic laboratories, medical labs, research purposes. It is widely used. So the last one is the traveling microscope. It is used for different different experiments in the physics laboratories. Traveling microscope. So the traveling microscope will give the precise optical instrument. It is a precise optical instrument which is used for measuring very small lengths and displacements. So it is used common in physics and optical instruments, as I said. So coming to the construction, it has a microscope tube. Here you can see it is a microscope tube. It contains lenses. It can be viewed moved vertically with the help of the screw. So with the screw we could able to move it vertically. Then the main scale, here we can see that the main scale, there are two main scales here. The main scale which is fixed on the base, okay, it is marked in either millimeter or centimeters. Then the vernier scale which is placed just above the main scale, okay, it will usually have 50 lines. Then we'll have the rack and pinion mechanism. In order to move the screws easy, you move the lens easily, we'll have smooth left and right rack and pinion screws, we can see here. And we'll have a glass stage where we'll place the specimen or the sample to view it. And the vertical scale is also there, vertical main scale and vertical main scale and vertical vernier scale. And we'll have a heavy base in which all this instrument is placed. So coming to the working of the traveling microscope, initially the object will be placed on this stage which is a glass stage, okay. Then the focus of one of the edge, like if you take a item over here, if you see, you will take the one of the edge and will observe the readings by placing the horizontal and vertical crosswire touching the edge of the object. We'll note down the readings like main scale reading and vernier coincidence will be noted out, and then we'll move to the other edge of the object. If in order to measure the displacement, we'll move to the other end of the object. Then also we'll be able to measure the main scale reading and the vernier coincidence. So after taking all these readings, we will be substituting in the formula called as total reading is given by main scale reading plus vernier coincidence times the least count. Here MSR stands for main scale reading, vernier coincidence stands, and times the least count. The least count of the traveling microscope is around 0.01 millimeter or 0.001 centimeter. It will have the number of lines on the vernier scale. Okay. Then where this traveling microscope is used? It is used to measure the diameter of a thin wire. As I said, it is used in optical instrumentary laboratories in order to measure the thin wire diameters. It is also used in capillary tubes. It is used to find the refractive index of a vertical displacement. It is also used to see the fringes or optics. It is also used to form a formation of Newton's rings, etc. So with this, we have completed the first unit, which is about the simple microscope, compound microscope, and parallel microscope, and how they are constructed, how they are built, etc. Thank you for listening.